Mr. B again, 19.1 bacteria. And it's interesting, I always do this topic second. I usually start with viruses first, as I stated in my virus video. We'll move into the smallest form of cellular life, specifically bacteria at this point here. I need to define a few terms, and they're pretty important. We need to differentiate the difference between a prokaryote and a eukaryote. Prokaryote is a very interesting name. Now, what pro in the name prokaryote means, let's be really clear here, pro literally means before there was. And karyo is a term that means nucleus. So the first forms of life, the most primitive forms, push that out of the way, there we go, of life that existed didn't have a nucleus. And it's important to understand these things are so small. Our cells, I always like to say a red blood cell is about 20 to 30 micrometers or millionth of a meter. These guys are so small that they're about the size of a, a nucleus of one of our cells. Okay, they're a fraction. They are a fraction of our red blood cells in size, for example. Our red blood cells, interestingly enough, start with nuclei, but lose them as they mature and make space for carrying oxygen. We'll get more into that in biology 12. So the unicellular organisms that we need to study are found all around us. They're found on us, in us, and in some of the most exotic and nastiest places in the environment you can even imagine. Because when we talk about the first single-celled life, and if we go back, literally go back beyond 2 billion years when these cells begin to appear on the scene, there's different camps of them. So we have to differentiate the domain, which is Archaebacteria, from the domain, which is bacteria. And that's where we go here. So I'll give you a chance to fill this in, take this in. But when you classify prokaryotes, we used to call them kingdom Monera. And that was just a catch-all phrase for anything that was a prokaryote. And that's it's still quite useful to think of it that way. I'm still sweet on that definition. Because when we put the prokaryotes in kingdom Monera, we just said it was all the single cells that didn't have a nucleus. But it turns out that's not sufficient. There's a lot more to it than that. What we have to look at is we have to look at these things now that we call domains. If you remember, we had kingdom, phylum, class, order, family, genus, and species for our classification scheme for everything on the planet. But when we were at the kingdom level, we sort of thought about that for a second. And we said, well, is there a higher classification than a kingdom? The kingdoms that you know would be kingdom Plantae, kingdom Protista, fungi, right? And we had a kingdom for bacteria, for Monerians, and then we said, hold on, let's break this down a little further, because we could look at similarities on critters on our planet and say, look, do you have a nucleus or don't you? And that distinction really, if you didn't have a nucleus and you were a cell, threw you into the Monerian camp. However, when we look at bacteria, there are very different bacteria. And on Friday, we did a really good job of zooming in, and we talked about the Archaea Bacteria. These would be some of the oldest bacterial types on our planet, amongst which we look at our halophiles. Now, philos is a Latin word which means to love. So let's differentiate this term. Halophilos and thermophilos literally means a love of, and we better change our marker or it'll never show up on the paper, salt-loving bacteria. And by salt-loving, we're talking about like the Red Sea or the Dead Sea, environments in which it's so salty nothing Let's make that salt loving. There we go. An environment in which something just couldn't survive, or environments that are so hot that it would kill the cells of just about any organism on the planet. But these ancient bacteria, I like to call them almost the hot tub loving bacteria. They not only love the temperature of something like a hot tub, but they would take uh, uh, a geyser, which could be beyond hundreds of degrees in temperature, just superheated water under pressure. Unbelievable. They love that heat. So we'll call these heat loving. These are some really old bacteria on our planet, and if we look in some of the most extreme regions on the planet, we can still find them. Modern bacteria, the ones that we all know and love, we look at, well, my favorite workhorse is E. coli, Escherichia coli, and you could look at Salmonella, and Bacillus anthracis. Ooh, I'm going to have to zoom out to make a little more room. The one that causes anthrax. Or the disease anthrax, to be technically correct. Zoom out. There we go. Now, when we look at our cyanobacteria, now the example that they're giving you here is just the blue-green bacteria, and these are bacteria that are heterotrophic. They eat other things. They digest our waste products that we leave behind. A way to organize this is to think about these two groups here as being the Monarans. This is the old Monarans definition as a catch-all. These guys used to be called the Monarans. But now we've put them in a much higher group. We say that we have domain bacteria, domain Archaea and everything else is a eukaryotic cell. So that would be any cell. We'll just do a little scribbling here that has a nucleus. So if you had a cell and it had a spot for organizing its genetic code inside of a membrane like that, then we say that you have a true nucleus. So it's important to differentiate the true bacteria, the eubacteria, from the archaebacteria. And there's very good reason for this. Eubacteria, they have a cell wall, but it's interesting and also very important to note that their cell wall is composed of a carbohydrate. We all, throughout high school, talk about the cell wall of plants over and over again. Plants have a cell wall made of cellulose, but plants are relatively young on the planet compared to bacteria. Right? Plants evolved in the last, say, 12 minutes on sort of our evolutionary clock. Bacteria were around for such a long time. Sure, they use a carbohydrate in their cell wall, but it's a very ancient one, and we call it peptidoglycan. There we go. So we'll just make that the cell wall. And it's their armor. It's very important. They need it. Without it, they're rather unprotected in their environment. And they're no dummies. They have their own form of protection. Now, E. coli, which is what we call a gram-negative bacterium, we'll get into the structures of different cell walls and their compositions when we look at different critters. And the classic comparison will always be Bacillus anthracis, which causes anthrax, versus E. coli. E. coli, as we zoom in and sort of take a peek at its structure, has a very smart way of protecting its cell wall. After all, the cell wall is what keeps the enemies out of the kingdom, right? Let's zoom in on a few things. First of all, we've got these little projections on the outside of bacteria that are called flagella. Now, what those are, they're not like human flagella, like what you would find on sperm. These things, instead, what these guys do is they whip, if you think of a prop motor, like on your boat, right? These flagella turn like a prop motor would, and they turn in one direction. So they're whipping. They're not whipping like sperm do in this eel-like motion. When you look at more advanced sperm, like you would, or advanced flagella that you would see on sperm, for example. No, that's not what you see. What you see is you observe that these turn either in this direction or they turn in this direction, like a prop of a motor, depending whether they want to go forward or back. So there's not that eel-like motion, which is much more advanced. We'll get rid of that. So flagellum is just for motion. That's the idea. Motion. And not all bacteria do that. Inside the bacterium, we see a region where there's the DNA, and I describe it to students. I'm like, imagine you just put yarn into, say, your purse or into your locker. If it went into your purse, which is my more favorite example, it would just kind of occupy the space it's in. DNA is kind of an interesting sort of snot-like substance when you actually extract it. And it's not in a nucleus. There's no, I'll try to draw one here. There's no membrane for this, right? This would happen in a eukaryotic cell. That's not happening in a prokaryotic cell. So our Archaea bacteria and our Eubacteria, they don't do that. And that makes them vulnerable in many respects. So their genetic code is here. Now, you won't see them with a genetic code like viruses. Viruses can do the single-stranded version called RNA, but once you get to bacterial cells and you move forward, the genetic code is DNA. So what's with these short, stunty little jobbies here? Well, I might as well just write it because it'll just help. The pili are for the transfer of genetic material. So when these guys have sex, they'll transfer a copy of a region of their genome through what you call the pili. So it's not really that incorrect to describe it as being the equivalent of being a penis. When you look at the use of it, if this cell were donating to another cell, I guess this one would be acting in a male fashion, or we say plus fashion, and the receiver would be female or the minus fashion. That's just a way of keeping track. What else do we see inside the cell? Well, one thing they didn't list is everything's floating around in this sort of thick goo that we call cytoplasm. And there are ribosomes in there, little protein factories, just like us. Zoom in a bit more. There we go. So what they've done, and just differentiate this. There we go. They've got that red region, which you're looking at. Let's see if they made a good differentiation of it here. They've put their peptidoglycan cell wall, and their peptidoglycan cell wall, you see, is sort of concentrated in the middle in that red region there. And an easy way to describe this to students, and I always say this is a good way to do it, is they've put membrane, cell wall, membrane. And that's a bit of a simplification, but it works. So membrane, cell wall, membrane. And they've basically bagged off their cell wall in a very interesting fashion. If you look at the region that I'm coloring here, you could think of the membrane as skin, if you'd like. You could think of this as skin, armor, skin, which sometimes I like to say to students, but understand that the armor is the cell wall and the skin is the membrane. This is now protected. And you say, well, why would it want to protect its armor? Well, we're coming after these things with antibiotics and drugs, for example, like penicillin. And if you hide away your cell wall, you're not as susceptible to environmental damage. Why not make the cell wall a little bit more impermeable? Bacteria like E. coli do this trick, and they're referred to as gram-negative bacteria because they do this. And when we identify them in the lab, we add different dyes, and these ones, for example, will turn pink when we do something called the gram stain. Oop, just undo that. There we go. So they're pretty smart, typical advanced bacterium, or a eubacterium here called E. coli. Now, E. coli are found in our gut lining. So if we do a little bit of drawing here, here is your large intestine. So as your small intestine releases, we'll go up the ascending limb of the colon in that direction. Draw a little arrow there. And then we kind of come across. And then it comes down. A little bit hard to see. But our large intestine is a region in our body where we have a lot of bacteria. And these bacteria here, these E. coli bacteria, just love to live in there. And why would that be? Well, I imagine it's because of all the food that we're providing them. And they're doing a job for us too. They're producing vitamins such as vitamin K, which is good for the clotting of blood. E. coli is a eubacterium that lives in a variety of environments. Having it live inside of our body is an advantage to us. Now, E. coli love to live in our large intestine. It's a very anoxic environment, very little oxygen, and E. coli prefers a low oxygen environment. A low oxygen environment for them is ideal. Now, E. coli will act considerably differently in an oxygenated environment. Heaven forbid that you get stabbed in the abdomen and they get into your blood, and then now you've got a real problem, because instead of E. coli living in your gut lining and doing this great job of living inside of us in a mutualistic way, we feed them, they produce vitamin K. Instead of that great scenario, now you've got them cruising around in your blood, infecting your organs and infecting wherever they would travel. And that's a great concern if you're ever stabbed. Heaven forbid that should happen. Interesting stat. Think of it, at least 70% of your feces, what comes out of your body, is your elimination of E. coli. They're almost like another tissue in the body in that respect. But they're very important. Interestingly enough, Archaea Bacteria, when we look at them, we think, well, wait, wouldn't modern eubacteria be descendants of Archaea Bacteria? It turns out not to be the case. They don't have any peptidoglycan. And when we sequence out their DNA, it turns out that they've got genes more like us than they do with eubacteria. So it's strange. They seem very distantly related to eubacteria, whereas we thought, well, they should have more in common with them. It turns out they don't. So the Archaea Bacteria, right, these cell walls, no peptidoglycan, that's odd. And to have their DNA sequences to be so different means we've got a lot more investigation to do on our Archaea Bacteria. We find these Archaea Bacteria in the most extreme of environments. Now, here we're looking at just this ridiculously nasty region. Hot springs, we could find them in acidic environments. We could find them down deep in the ocean, and these things that are called black smokers, living off of the chemicals that are being produced from deep within the earth. Sulfur compounds. And it's a little bit hard to see from the picture, but there are these streams of snot, and in these little streams, this is a sort of my animation of it. Inside of those streams, or those little globs, are the bacteria living in these little colonies. and they are nibbling, making energy from compounds they're coming in close contact with. You got to think about this. There's no other nutrients around for them to consume. They're breaking the bonds of sulfur compounds so that they can survive, so that they can add to their own respiration, their own metabolism. So amazing what goes on down deep in the ocean. And Archaea bacteria, which would have been the very first cells on the planet, are found in extreme regions on our planet. It's like they're living in regions where they would have developed at the very beginning of time, or at least their time. So when you're faced with this task, imagine if I walked in a room and I said, okay, here's your job. Here's all the prokaryotic or bacterial cells on the planet, and I'll be back in, a month or two. You tell me how you divided them into camps. Remember, the biologists are we're grand librarians of nature. We not only try to determine how things work, but when we determine how things are related, we set up these structures and try to figure out, okay, what camps would we put organisms? And we've done that. We've got bacterial camps and plant camps and protist camps and fungal camps. You get the idea. And we came up with a couple of simple characteristics for identifying bacteria. A couple ways we can do that. Method number one, we can identify them based on their shape, and I'll go over that in a minute. We can look at what the compounds are that make up their cell walls, check out that too, see what kind of structures they have for moving, if they move at all, and then we look at their metabolism. How do they get energy, right? Do they eat other things? Are they heterotrophs? Are they autotrophs? Can they make their own energy through photosynthesis? Or are they what we call chemotrophs? Can they produce their own food by breaking down chemicals in nature? So let's look at these criteria as we move forward. This is testable. As soon as we start to apply a model to determine how we should organize things, it's got test written all over it. Okay? Very important schematic or structure that you need to know. Criterion one, if we look at their shapes, now we'll elaborate on this more because there's not only shapes but groupings. Rod-shaped cells are known as bacilli. Now, rod-shaped, here we go, is just a loose term. Slightly rectangular with rounded corners. You get the idea, like a rod. Okay? Or cylindrical if you want to think of it that way. So when we look at that, we say, oh, we're looking at bacilli. And this is characteristic of E. coli. E. coli looks like this. But that's only one characteristic for identifying them. You might have heard me say gram-negative earlier. Well, that's another important test we use to help group these guys. Now we had sort of cylindrical or rod-shaped ones. Now we have ones that are shaped like a ball, and we say that those are coci. Sometimes you'll hear some people pronounce it cocci. It doesn't sound quite as elegant. But spherical prokaryotes, here you go, coci. And later on, we'll talk about what it's like when they get together in groups. This, just so you know, would be a diplococci, them hanging out, sort of together in as best friends. And if we look below here, we can see them forming these sort of long chains, sort of like a necklace. And we'll get into it more, but this is referred to as a streptococcal association. Sure, they're all cocci, but strepto means that they're forming chains. So there you go. You got bacilli, or rods. You've got spheres, so rods and spheres, coci, and you need to know the names. You can't just say, well, it's a sphere. That's not good enough, right? Official terms. And then we've got one like this. And the first one that comes to mind, always for me, is the syphilis eubacterium. Now, its name more or less gives it away here that this is spiral shaped. And there's that spiral pattern. As soon as it does that, we have yet another genetic or generic shape to refer to. So we've got rods and spheres and spirals. And syphilis, yeah, nasty disease. Obviously, one of the worst sexually transmitted diseases. If those guys get into your bloodstream, migrate their way up to your brain, yes, very nasty result. Another way that we can classify bacteria is based on the composition of their cell walls. Now, there's many tests to do this, but one of the most fundamental tests is a test that we call the Gram stain test, or shorter, the Gram stain. And the Gram stain is usually, sometimes you see it in high school biology, other times you see it your first time in college. But what is done with the Gram stain is we apply two types of dyes, one that's violet and one that's red. And we see by the time the staining tests are done, what color did the bacteria absorb? Well, gram-positive bacteria are positive, and they, by a positive test, we say that they retain the violet color. They're positive for a dye called crystal violet, but I'll just refer to its color as being sort of a purplish hue. Ooh, violet. Let me spell properly. It is getting late. There we go. And you think of it, it's a darker color. Sometimes it can be hard under the microscope to identify. It's just much darker. And what it means is that their thick exterior cell wall, they wear their cell walls on the outside. They're not as smart as E. coli, which kind of bagged their cell wall off with a little bit of membrane on the inside and outside. No, gram-positive bacteria, they're just like soldiers. They've got their armor on the outside, and they're using that to protect them from the world. We'll zoom in. This here is a gram-positive bacterium, and what we're talking about, what we're referring to, is the Peptidoglycan layer, which is, let's choose a new color because I'm getting a little tired of that one. Here, runs from this here is the inner membrane, this region, and its armor. This is just like a soldier, right? There's the skin or the membrane, if you want to think of it that way. And they got this very thick exterior cell wall made of this material here, peptidoglycan. This is something that an antibiotic like an ampicillin or a penicillin, etc., is very effective at disrupting. And it has worked for a very long time affecting these bacteria. However, bacteria evolve, they have very quick life cycles. They can reproduce in many times in under 20 minutes. The ones that are positive for the gram stain turn purple. So let's make that distinction. You see this color here? Purple, or very, very dark color. Dark shade of, well, I wouldn't say red. I'm more comfortable with the purple. Gram-negative bacteria, they have their membrane surrounding their cell wall. So there's membrane here, and there's membrane here, and their cell wall is here. And the gram stain can't really get at it. It can't get there. So it doesn't stain it the same way. These ones take what's called the counter stain. They take safranin, and I'll spell that for you, and they'll appear sort of a pink color, a light pink color, whereas these will be dark or purple when you look at them under the microscope. So we could say we've got gram-positive bacteria, gram-negative bacteria, and we start to put these names together. Gram-negative bacilli and gram-positive cocci, and you can see how these classification schemes come together. Gives us a little bit of a better idea about what we're dealing with. But the gram negatives definitely have that. It's very thin armoring. It is, but it's tucked away, and they've managed to protect themselves quite well with it from the environment. Another way of classifying bacteria is based more or less on their metabolism. How do they get energy? How do they survive? What do they do? So the metabolism. And there's heterotrophs, and that's like us, right? We consume other organisms to survive. And then there's autotrophs that can make their own food from inorganic molecules. Okay, photosynthesis is the classic autotrophism, where you take carbon dioxide and you take water and you take some energy from the sun and a few minerals for good measure. And essentially, and it's interesting how some folks put this together, some don't, but what's produced if you make your own food using the energy of sunlight is a carbohydrate. See? Carbo and water, right? When we drink, we hydrate. C6H12O6. I always like to point out to kids, students, what is this reduced to if you look at that molecule? 2 parts hydrogen to 1 part oxygen? And that kind of reminds you of H2O to the first. If you divide those down by 6, you get the idea? Carbohydrate. That's where the actual name came from. Now, plants are some of the greatest autotrophic organisms on the planet. Now, there is chemoautotrophy, which is another layer, and sometimes we just call them chemotrophs, like those Archaea bacteria way down deep in the ocean that can use sulfur to make their own energy-rich compounds. That is autotrophy, but to be correct, we'd call it chemoautotrophy. A term comes up here where they talk about the process of cellular respiration, and what does that really mean? Well, when you respire, it's kind of like a simple way of thinking about it. Cellular respiration, big scary term, but it means a cell that takes in its nutrition, and you can kind of think of that as like gas, and that nutrition has to be turned into all the energy. It has to all those food compounds wherever they come from, whether it's glucose or whether it's some kind of sulfur compound. It has to be broken down. It functions basically like the fuel. And for metabolism, what you do is you run that fuel. Now in our cells, we run it in the mitochondria, and we use it. Now I'll just say this in eukaryotes, to make energy. And that's, I do this example with eukaryotes. I know we're talking about bacteria here, but you put the gas into the generator, and the generator produces the electricity that runs the house. Okay, so that's just a rough analogy. Now, that's a neat way of erasing, undoing about 1,000,000 times. There we go. If a bacteria takes in energy-rich compounds, it's going to use them as well. It's going to break them down. Now, it'll break them down in a way where it releases energy. So it'll just take in the energy-rich compounds, and it'll use things like enzymes to produce energy. And in fact, one thing that we know is that mitochondria are exactly where this occurs in our cells. Mitochondria are aerobic bacteria that are very good at doing this exact thing. They're trapped aerobic bacteria, which live with us, and they produce our energy in spades. But we talk about that in detail in Biology 12. You can do cellular respiration, and that's, you think about it, when the gas powered generator runs, it's in the presence of oxygen, and if you if you don't supply oxygen, the generator can't function. But there is another way of generating energy, like what happens when you feed a lot of sugar to, well, let's put it this way, how you make wine. You can feed a heck of a lot of sugar or energy rich compounds in a low oxygen environment, and it's harder to make energy without O2, but it could still be done. The production of alcohol uses fermentation, and it's not nearly as efficient. Cellular respiration is the champ, but fermentation is a very quick way to produce energy, and it can do it in an absence or very little oxygen. So there's two kind of pathways there. You've got this one, and you've got fermentation, where alcohol is the byproduct. Now, just how much oxygen do you think certain critters on our planet like? Now, obligate is an interesting term. We've got obligate aerobes, and what that means is, I like to say obligate, and you say, well, obligate, is that, what do you mean, obligated? Yeah, they kind of have to be aerobes. They require oxygen. So let's just highlight this region here. And a classic example of this one, not a very nice critter to talk about, let's change colors, I want to go purple, is you could think about Mycobacterium tuberculosis. Tuberculosis targets the cells of the lungs, and it's a very horrible infection. These are obligate aerobes. They love and demand to be in the presence of oxygen to survive. They need it. Their metabolic processes just won't function without it. But there are bacteria which are obligated to not be in oxygen. Okay, so this group is the no O2 group. Okay, so death to O2, absolutely not. And this is the group up here, has to have it, has to have it, has to have it. They'll die without it. Obligated to be anaerobic, let's circle that an, that means without. Critter that comes to mind for this is when you look at something like, reminds me of a friend of mine one time, and it also reminds me of the Civil War. In the Civil War, you would get gangrene, and the bacteria that cause gangrene, those bacteria can absolutely not handle oxygen. They just hate it. And a lot of people in the Civil War, what was happening was there would be shrapnel, there would be bits of filth embedded under the flesh, bits of dirt, etc. And some of these germs that get underneath the flesh, that's actually very low in oxygen content, they'll grow there and they'll cause gangrene. So the same critter that causes gangrene is an obligate anaerobe. So let's write that down. The critter's name isn't so important as the term, because I bet you've heard of gangrene before. And if you haven't, what happens with gangrene, not to freak you out too much, let's pop out to the web. If you get gangrene, what will happen is there'll be a bubbling under the skin. It's very nasty. So not the nicest of things to get. And if you develop an infection this way and you get this green sort of bubbling under the skin, it's kind of a mode of last resort to remove the limb. By today's standards, what you could do is you could introduce oxygen under extreme pressure, like in a hyperbaric chamber. And not, I guess extreme's a bit too much of a term, but by introducing oxygen and pushing it into the tissues, you could actually kill these bacteria. Okay? So what do we have here? We had the ones that were obligated to live in oxygen, the ones that were obligated to stay away from it, and then we had the ones with the faculties to deal with it. Now this is like E. coli. Let's switch up the colors. E. coli has the faculties to live without oxygen. So if you say it's facultatively anaerobic, it would prefer no oxygen. But can deal with it. And a classic example of that is E. coli. They're so wonderful in our bodies, in our large intestine, but I really wouldn't want E. coli getting into my bloodstream. A very oxygenated environment, they're much different in an environment like that. So, they have their preferred environment and their non-preferred environment. So you've got obligated aerobes, obligated anaerobes, and you've got the ones with the faculties to do sort of either thing. So I guess it is fair to say you could have a facultative anaerobe, but you could also have a facultative aerobe, one that could handle no oxygen but would prefer to be aerobic. Now, when bacteria reproduce, they can reproduce extremely quickly. Now, sexual reproduction, if you think about it, takes a bit of time when bacteria transfer genes from one to another through the sex pilus. Now, bacteria would do that to create variation within the population. Generally, sexual reproduction occurs when a population is trying to, as I've said, increase its diversity, right? Tough times, sexual reproduction helps out with that. But asexual reproduction is sort of like a moment of opportunity. If you leave some spaghetti sauce out on the counter and E. coli encounters that, it'll put sexual reproduction on hold and just clone itself like mad because there's such a wonderful opportunity in the environment that it clones and goes after that food source, and it leads to food spoilage, of course. This is just cloning. So just make a little note: cloning. Now that is not good for genetic diversity. In fact, this asexual reproduction doesn't introduce any at all. It's just the cell replicating like mad. And bacteria can do this. In Mathematics 9, we were looking at formulas, exponential growth of bacteria, because they'll go into this reproductive cloning phase so quickly. And what they do is they produce daughter cells. Interestingly enough, they're just clones. And we clone cells all the time. If you cut yourself, the cells grow in by mitosis, right? And they're just clones of the cells that were there before. So it's a quick way to reproduce, and it's just a survival strategy in nature. Helps you to take advantage of your environment. So there we go. Identical daughter cells. And we refer to this as binary fission because it splits into two. And fission means to split. And that's the great idea there. Conjugation is when one bacterium decides to have sex with another. So here's a sort of an enhanced diagram, if you will, of one bacterium transferring genetic code to the other. So this hollow bridge, we saw this before. Let's switch colors to something that's a little more evident, right? This hollow bridge is a pilus. So through that, genetic material would be transferred. So at this level, we just say that genes are being sent. Now that's the genes and how they're sent. The genes are copied. Okay, so gene copies. Some of the genetic code will be sent over, and interestingly enough, there's extra little bits. They don't show this very well, but there are these little things that are called Plasmids, they're like extra bits of genes that are exterior to the main, let's say this, let's zoom in, your main genetic information might be here, but you might have these little things. They're like coins, and we refer to them as plasmids. Those can actually be copied and sent over as well. And some of these plasmids can be downright nasty because these extra little bits, oops, have to, these extra little bits of genetic material are sort of transferred, like I've been referring to as coins. I'm going to change the color to green, but I'll go back. And they can have some pretty nasty genes. And some of these genes are for antibiotic resistance. And we find that bacteria like hot swapping these genes, and they're staying ahead of us. So when we use antibiotics against bacteria, what we're really doing in a Darwinistic way is we are killing off the weak bacteria, and the bacteria that got genes through sexual exchange or just lucky bacteria that mutated and developed resistance, those bacteria are surviving, and the survivors who are strong are reproducing, and our antibiotics aren't working as well anymore. So there's extra little bits of genetic material you have to be aware of, not just the DNA, not just the DNA, but these little bits that we call plasmids. And boy, do they get hot swapped. Because bacteria are doing their best to survive. Some bacteria can even form spores. This is amazing. When you look at anthrax, it will do this. If times are so bad that there's very little food in the environment and you're either going to die or into a state of suspended animation, that's when some bacteria, such as Bacillus anthracis, better known for the disease anthrax, let's just write that. What they'll do is they'll form this little thing called an endospore. And they literally dehydrate themselves, and they create this portion in a cell called the endospore, right there. And it is kind of like the name says, it's emphasis on spore. It will lie dormant in the soil for extreme amounts of time until, well, I don't know, it's moist and wonderful and there are food sources back again, and then it will reverse course and grow back out into the full anthrax critter, the full bacterium, once again, and take advantage of the environment. So endospores are nasty because they've been used in they've been used in biological warfare. Whenever you hear about a consulate receiving a white powdered envelope, panic hits for a very good reason, because have these bacteria been used as a way to attack our politicians, do something nasty against somebody else we don't like, or that we just don't agree with? According to the Geneva Conventions, this doesn't happen because biological warfare, it's over the top, it violates the rules of war. It should not be happening. Finally, kind of get away from the serious topic, we say, well, what are bacteria good for? What do they do for our world? Well, they produced our atmosphere. We should thank them for that. They were the first photosynthesizers. Go back a couple lectures, we talked on and on about 2 billion years ago and the production of oxygen as a waste product, which without oxygen, our mitochondria in each of our living cells wouldn't be able to run like a gas-powered generator to produce energy for us. Cleverly, critters on our planet adapted to take advantage of all the energy that was captured from the sun in the form of photosynthesis. Other bacteria, well, If I look out into the forest, all those leaves have hit the ground, and it's not just fungi that are decomposing them, but bacteria. They're just crawling all over them, breaking them down, and turning dead matter into their food source. But without them, can you imagine the leaf litter in the forest? It would just we would just it would be overwhelmed. It would just stack up year after year. Humans, though, have taken a bacteria for many other uses. Very handy little critters, as it turns out. First one that I think of, and you say thank you, is E. coli in your gut lining. Very, very handy. We're learning to even use them now. We're programming their genetic code to make to help us make proteins and our insulin, and even to produce chemicals that we need to survive. However, the food and beverage industry, right? We have to be aware. When we look at food and beverage, I kind of look at bacteria and I say, well, you got to be aware that there's a great cost incurred by bacteria on foods and beverages. We're doing all sorts of things to preserve our food. Once upon a time, we would salt pork, or we would make things, we would can things in sugar content so high that the bacteria could never live on it. They would get dehydrated, right? We can use bacteria to remove waste and poisons from water, mine things from the ground, as I've said, synthesize drugs and chemicals, and vitamin K specifically in the human intestine. Heck, they even help us produce our food and beverages. Pretty interesting stuff. We need them, they need us. And ladies and gentlemen, that is more or less it for 19.1 bacteria. That means what remains is a discussion on disease, and no discussion of disease would be complete without talking about immunization. Okay, so have a good one. That's 19.1 bacteria. Most important thing, not to make you dizzy here, is to realize that we had looked at many, many ways of identifying bacteria. Kind of going back to these criteria, and then we did question number 4, I wanted to point out, is all about their metabolism. And that's when we got talking about anaerobic, aerobic, and the various forms of that. Okay, ladies and gentlemen, have a good day, evening, or morning, whichever it may be.